Mrs. Chairman. In order to balance, I will talk in French. Merci beaucoup au réseau Brûlé Ulcère de nous avoir invités pour présenter aujourd'hui notre modeste expérience sur la prise en charge des mycétomes au Sénégal à travers des laboratoires, un laboratoire de référence et des laboratoires satellites. Alors, comme vous le savez, Wendy en a parlé hier, les mycétomes représentent vraiment l'une des plus négligées maladies parmi les maladies tropicales négligées, donc qui atteignent surtout les pauvres des pauvres. Quand on a fait une petite revue de la littérature, on se rend compte qu'en Afrique de l'Ouest, sur la base des cas qui ont été corrigés, le Sénégal reste le pays le plus endémique. Alors, elle a parlé hier un peu de la difficulté de la prise en charge des mycétomes liée à la multitude d'agents pathogènes rencontrés. Ici, il s'agit d'une étude qu'on a faite au Sénégal il y a 2 ou 3 ans pour faire un peu le report et la revue des cas observés au laboratoire. Nous avions à peu près 47% de formes fongiques qui sont plus retrouvées dans la zone nord et à peu près 36% de formes actinomycosiques qui sont un peu distribuées dans le reste du pays. Et toutes ces espèces ont été retrouvées un peu dans les cas qui ont été corrigés, notamment les grains noirs dus à Madurella mycetomatis et à Falciformis spora senegalensis, Actinomadura pelliteri qui est très prévalente lorsqu'on compare un peu avec ce qui se passe au Soudan, et enfin toutes les autres espèces qui peuvent donner des grains blancs comme Actinomadura maduré ou Streptomyces somaliensis. Donc on peut voir les différentes formes de mycétomes observés. Alors, la prise en charge est vraiment cruciale et différente selon qu'on ait des cas d'actinomycète ou bien des cas fongiques. Par exemple, lorsqu'il s'agit d'une actinomycose, on traite avec du cotrémoxazole. Par exemple, ça c'est une de nos patientes qui a un mycétome du dos. Quand on a traité, il y avait une très bonne, un bon succès thérapeutique. Par contre, quand il s'agit des formes fongiques, c'est beaucoup plus difficile parce que, en plus du traitement médical, à savoir l'itraconazole et la terbinafine, il faut faire de la chirurgie parce que le plus souvent, on a une atteinte osseuse. Donc, vous voyez, pour ce patient-là, malgré le traitement, on a été obligé d'amputer le bras. Donc, pour dire qu'il est extrêmement important de poser le diagnostic correctement. C'est la raison pour laquelle, au niveau du laboratoire, on a plusieurs techniques. D'abord, le challenge, c'est d'obtenir les grains. Sans grains, pas de pathogène. Ça, Wendy en a parlé hier. Alors, il y a la possibilité d'avoir les grains à partir des sinus, mais on a montré que cette technique de sampling n'est pas très intéressante parce que les grains ne poussent pas. Donc, la culture est négative. On peut faire de la biopsie. Jusqu'à maintenant, nous, on faisait de la biopsie en utilisant ces punches qu'utilisent les dermatologues. Mais ça ne marche pas très bien parce que lorsque la lésion est profonde, lorsqu'elle est superficielle, on peut avoir des grains, mais lorsqu'elle est profonde, il faut aller carrément prendre les grains lors de la chirurgie et c'est difficile. Et là, actuellement, nous sommes en train de suivre un peu ce que font l'équipe du professeur Fahal, à savoir l'aspiration à l'aiguille fine. Alors, la première étape a été de se faire former. Donc, grâce à l'appui de l'Organisation mondiale de la santé, nous remercions Augustin, Kingsley et toute l'équipe qui nous a supportés pour aller au Soudan, apprendre ce qu'ils font sur l'ultrason, sur la paillasse, sur comment faire une deep biopsie, tout ça, on a pu l'apprendre. Et une fois au niveau du Sénégal, nous avons essayé de refaire la même chose, c'est-à-dire de maintenir d'abord les 2 techniques, à savoir la culture sur milieu de Saburo et Lowenstein en fonction de l'espèce actinomycosique ou fongique, la cytologie, mais surtout, on a essayé de mettre en place la technique de biologie moléculaire. Alors, ce que nous avons fait, c'est que nous avons regardé un peu tous les laboratoires qui se trouvent au niveau du Sénégal. On a essayé de faire un petit survey pour voir quel était le plateau technique disponible. Et on a choisi 2 laboratoires satellites. Un laboratoire à Dakar et un laboratoire à Thiès, avec le minimum d'équipement disponible. Et ensuite, nous avons essayé de mettre en place l'unité de biologie moléculaire au niveau de la zone nord. Donc là, je donne un exemple d'un cas, comment on prend en charge un cas. Donc c'est un patient de 39 ans. Lorsqu'il est venu à peu près 2 ans d'évolution de la lésion, on fait d'abord l'échographie et la ponction à l'aiguille fine. Et après avoir fait l'échographie avec ponction à l'aiguille fine, on peut voir qu'à partir de l'échographie, on peut dire est-ce que ce sont des grains qui sont actinomycosiques ou fongiques d'abord. Et ensuite, on recueille la pièce de biopsie et surtout, on identifie les grains Et c'est les grains qu'on pose, par exemple là c'est un milieu de Lowenstein qui nous permet d'avoir l'espèce. Okay? Ici ça pose pas de problème parce que c'est Actinomajora pelliceri, mais après il faut recourir à la biologie moléculaire. Alors pour la biologie moléculaire, on a envoyé une étudiante. Oui.
Il y a la traduction.
Il
faut parler un peu plus lentement.
Lentement, ok, excusez-moi. Alors pour le laboratoire de biologie moléculaire, nous avons envoyé une de nos étudiantes chez Wendy pour se faire former sur les techniques de biologie moléculaire. Et après, elle a transféré la technologie à Dakar. Et parallèlement, pour pouvoir optimiser nos techniques, nous avons également mis en place une biobanque. Donc, toutes les souches cliniques qu'on collecte, on les garde au laboratoire pour pouvoir faire cette biobanque. Dans le cadre d'une étude qu'on est en train de réaliser avec les collègues dans le cadre du projet JORAT, nous avons essayé de colliger des cas. Et lorsque nous avons colligé un certain nombre de cas, nous avons testé un peu nos plateformes sur ces pathologies-là. Bon, par exemple, ici, on avait 67 patients qu'on a recrutés. Et pour ces 67 patients, on avait à la base que 17 patients qui étaient revenus positifs après culture. Pour dire que là aussi, la culture est difficile à faire lorsqu'on est au niveau du terrain. Donc ça, ce sont un peu la répartition des cas qui ont été rencontrés. Mais surtout, on a essayé de mettre en place la biologie moléculaire, donc avec la technique d'extraction. Avant, on utilisait la technique du CTAB, mais les premiers tests qu'on avait réalisés n'étaient pas concluants. Donc on a été obligé de prendre les échantillons, de les envoyer chez Bondi pour pouvoir faire la confirmation en se basant soit sur le gène CZ pour les actinomycètes, soit sur le gène ITS pour les formes fongiques. Et lorsqu'on a envoyé et on a reçu les résultats, ça, ce sont les résultats de l'histologie qu'on faisait à Dakar. Et ça, ce sont les résultats de la PCR qui nous a été rendue après par Wendy. Et on voit que pour certains cas, on se rapproche un peu de ce qu'on avait en histologie. Mais pour d'autres cas, c'est la biologie moléculaire qui a su réajuster l'agent pathogène en cause. Par exemple, ici, Madurella mycetomatis, c'était plutôt du tropicana. Là, on n'avait pas pu identifier, mais c'était du Gordonia terrier, etc. Donc, la biologie moléculaire est extrêmement importante pour pouvoir confirmer. Et enfin, certaines espèces ou nouvelles espèces ont été identifiées dans le cadre de cette étude-là. C'est le cas de Madurella tropicana, de Madurella pseudomycetomatis, qui n'ont jamais été décrits auparavant au Sénégal. Donc, du coup, le fait d'utiliser la biologie moléculaire peut facilement améliorer le diagnostic, mais surtout après, lorsqu'on prévoit de faire un peu le suivi de la résistance aux antibiotiques. Donc, je terminerai par dire que la collecte des échantillons est difficile, c'est challenging, mais on peut réussir à le mettre en place, notamment avec l'échographie, mais surtout avec la ponction à l'aiguille fine. Il y a un manque de coordination entre les spécialistes. C'est extrêmement important si on veut bien prendre en charge les mycétomes. Et surtout, il faut former les gens sur le terrain. Il faut informer le ministère de la Santé. Et c'est une excellente opportunité que nous offre le Skin NTD Network pour pouvoir continuer à travers sur cette pathologie. Nous allons terminer par remercier la Fondation Dioraft qui a financé une bonne partie de nos recherches. Le centre MRC, vraiment le professeur Fahal et Wendy qui nous ont beaucoup aidés à pouvoir réajuster nos diagnostics, le ministère de la Santé et toutes les universités qui ont participé dans ce projet. Merci beaucoup.
Merci professeur Doudou pour cette belle présentation. S'il y a des questions, some questions, professeur.
For your presentation. I'm not used to mycetoma, but I'm interested in it. Because there are so many species involved in the transmission, we have the bacterial, fungal, and other species that we are yet to identify. I would like to know if it's possible to do direct DNA sequencing rather than using different primers for amplification for PCR. Is it possible to do direct DNA sequencing for species identification? Thank you.
Right now, that is what we are doing with Wendy and Professor Farrell. We are doing direct sequencing, targeting two genes, the 16X genes for the actinomycetoma. So we do extraction and we do sequencing. to identify the pathogens. And for fungal, we target the ITS. But to facilitate the screening in the field, Andy and Professor in the group is developing real-time PCRs and real-time PCR to screen rapidly the pathogen in the field. Yeah, Wendy.
So what you asked was if sequencing directly on the grain would be possible too. What Professor Verhaal's team together with CDC demonstrated that the grain typically only contains less than 1% of the pathogen DNA. The rest is human DNA, so it was for them very difficult, almost impossible to identify the causative agent just by sequencing. But the deep sequencing you could identify probably, but you cannot get a whole genome out of it. So I think that pan-fungal may be better.
Isra?
Oh, Professor Farrell. Professor, I give you the word.
Thank you very much. Thank you very much, Dodi, for this nice and elegant presentation. It is very clear. It is a matter of training of staff to be able to identify the organisms. So I hope this network will help you and help us to work together so as to train as much as possible of the staff around Africa, not in Senegal alone. So they can be able to identify. Cases are there, but nobody knows what is happening to them. So we need a proper training for our staff to be able to identify these cases and to bring them for early cases, early management and treatment. And thank you very much.
Isra, first.
Just a curiosity about the status of research on serology, let's say detection of antibodies or cell-mediated immune response in the diagnosis of mycetoma. I don't know if that's a field that has been abandoned or is some research going on. Just curiosity.
In Senegal, not yet, because we are on the starting point, but I think Professor Fahal or Wendy have done some work on this.
Serology, not very much a fascinating investigation in our country. It's tedious, take a long time, you need to prepare the antigens, and as you know, there is more than 80 causative organisms for mycetoma. So it is very difficult to find the antigen that contain all these 80 organisms. This is the one problem. It takes quite a long time, and not very specific, and it's not very useful, the serology at this side. The immunology, you are working on it, a lot of promising results coming, but there is no shouting result from that. It is almost within normal pattern, the immune responses of this patient. And this is the problem. This patient has a good immune system, and in spite of that, they're developing this mycetoma. It may be genetic, it may be environmental, it may be other causes of that side. Thank you.
Also for this comment.
In addition to that,
yes.
There are recombinant antigens for the main causative agent, but we didn't find any, well, we did find difference in antibody levels against these antigens, but not a clear-cut yes or no. So many of the healthy endemic controls also have antibodies against the identified antigens. So as far as I know, there's currently no active search for a new antigen because of that.
Wendy, Professeur Kadima.
Ok, merci. Moi, j'aimerais féliciter le Sénégal parce qu'en une année, vous avez fait un pas de géant avec les résultats que vous montrez, mais mon inquiétude reste toujours en amont. En amont, vous avez des atouts. Un programme de maladies tropicales négligées qui fonctionne et qui peut être appuyé pour que la détection se fasse de manière intégrée et que vous puissiez maintenant encore davantage bien cartographier la situation du Sénégal et profiter, parce que vous avez dit que vous avez corrigé vos données. Est-ce que, je n'ai pas bien vu, en dehors de la discordance sur 2 cas que vous avez mentionnés, est-ce que du niveau clinique, de suspicion clinique, est-ce que vous êtes très rapproché de la réalité ou pas? Parce que ça, c'est intéressant du point de vue épidémiologique de voir, comme on le dit, il faut qu'on forme les gens. Il faut que les acteurs de terrain soient les plus bien outillés pour pouvoir commencer par suspecter afin de prélever l'échantillon et vous envoyer. Donc, c'est cette collaboration avec le programme national. Il faudra vraiment l'encourager. Nous sommes là pour essayer un peu de vous accompagner là-dessus. Merci.
Merci beaucoup, Dr Kadima. Je pense que ça, c'était un peu la première étape qui va aller vers cette collaboration-là. Comme vous l'avez dit, il fallait d'abord qu'on se fasse former sur les techniques de référence utilisées pour un bon diagnostic des mycétomes, ce qui a été fait en collaboration avec le Soudan et Rotterdam. Maintenant, la deuxième étape, justement, après avoir mis en place et qu'on a commencé un peu à confirmer correctement les cas, la deuxième étape, c'est d'aller discuter et travailler avec le programme pour justement essayer de détecter les cas au niveau du milieu communautaire. Comme ça, lorsqu'ils vont remonter les cas, nous serons à même de donner un diagnostic le plus fiable possible. Merci.
D'autres questions? Oui.
Merci beaucoup pour votre présentation. J'aimerais savoir, est-ce que c'est possible de faire l'identification des espèces par la microscopie? Est-ce que ça pourrait être fait dans les zones périphériques en attendant de les référer au centre?
Alors, par microscopie, c'est très très difficile de différencier, pratiquement impossible de différencier les espèces. Il y a la couleur des grains qui permet d'orienter, parce qu'on sait que par exemple les grains rouges sont dus à Actinomadura pelletieri. Parmi les grains noirs, il y a plusieurs espèces. J'ai montré tout à l'heure, on parlait avant de mycétomatis, on parlait de falciforme spora sénégalais, mais il y a d'autres espèces qui sont là. Et la microscopie ne peut pas permettre de pouvoir différencier les agents pathogènes en cause. Merci.
Des commentaires? Oui, professeur Farah.
We can tell you this is microscopy can tell you this is bacteria or fungus, and that is it. So you can have a broad line. This is bacteria or this is fungus, and then you can decide. In the field, you have no other facilities. Thank you.
Question? Other question? Donc, on va remercier le professeur Doudou pour la présentation, pour les questions. On l'applaudit. Donc, nous allons passer des mycétomas à Yos, Pian. Demander à Dr. Isaka Maman, molecular detection of Treponema pallidum, responsible for skin ulcer in people suspected of Yos in Togo. Donc, Dr. Maman, vous avez la parole, 10 minutes aussi.
Merci beaucoup. Nous remercions le BU-LABNET pour nous avoir invités à cette réunion et nous donner l'occasion de faire cette présentation qui sera faite en français. Donc, en gros, je vais vous parler de la détection moléculaire de Treponema pallidum qui est responsable des ulcérations cutanées chez la personne suspectée des pieds au Togo. Donc, voici le plan. Rapidement, on passe. Alors, retenons que le Treponema pallidum est le pathogène en cause des ulcérations qui sont observées chez la personne suspecte de pion. Alors, cette maladie tropicale négligée est prédominante dans les régions tropicales d'Afrique, d'Asie, d'Océanie et en Amérique du Sud. Au Togo, les premiers cas ont été diagnostiqués entre 2019 et 2020, dont 32% ont été confirmés par le test d'IPP. notamment dans 3 districts. Alors, habituellement, les techniques de diagnostic reposent sur les examens cliniques et l'utilisation des tests sérologiques, le test rapide ADT et le DPP. Alors, dans le cadre de l'éradication de la maladie, il a été recommandé d'utiliser le test PCR qui permet de supporter le programme de cette éradication. Alors, le Togo, dans son plan stratégique, a inscrit l'éradication du plan à l'horizon 2030. Donc, ceci nous a amené à mettre en place la technique moléculaire PCR pour supporter leur programme, ce qui fait l'objet de cette étude. Alors, l'étude a été menée dans 3 districts au Togo, notamment dans ce district, dans la région des Plateaux, dans le district de Mo, dans la région centrale à la frontière avec le Ghana, et dans le district de Toul, qui est à la frontière du Burkina Faso. L'organisation de la collecte des données ainsi que les échantillons est assurée par le programme qui effectue chaque année des campagnes de sensibilisation dans les villages ou dans les écoles. Ceci en collaboration avec notre institution qui est le laboratoire national de référence. Et dans ce cadre, 3 campagnes ont été organisées, notamment en 2021 en juillet, en décembre, en 2022 et cette année, de mai à juin, dans 3 districts précédemment cités comme endémiques. Par rapport aux échantillons collectés, il s'agit du sang, notamment par ponction au niveau du bout du doigt pour les tests rapides et l'écouvillonnage en utilisant les écouvillons à polystyrène pour les tests PCR. Globalement, l'algorithme qui est suivi au pays, qui est aussi recommandé par l'OMS, donc les premiers cas sont testés avec les RDT. Lorsqu'ils sont positifs, on fait les DPP. Donc les échantillons qui sont collectés pour la PCR se font sur les cas positifs au DPP. Alors, l'analyse moléculaire se fait à l'INH, qui est l'Institut national d'hygiène, qui est le laboratoire national de référence pour les maladies à potentiel épidémique au Togo. Et globalement, nous travaillons sur les maladies virales, notamment la grippe, les fièvres hémorragiques virales, les arboviroses, la COVID-19, les méningites bactériennes, bien entendu le C de Brûlis, dont nous sommes le laboratoire national de référence, le PIAN et l'onchocercose. Alors, par rapport à la détection moléculaire, ce qui commence par l'extraction, on utilise le Genolyze Kit, après l'avoir validé sur le Kiagène. Et par rapport à la détection moléculaire, nous utilisons le comptable bioperfectant multiplex Sopamix, ainsi que les primers et les probes pour les gènes. Et notamment, nous ciblons ici les gènes TP858 et le gène Pool A. Alors, la plateforme en termes de machines utilisées, c'est le Quant Studio. Par rapport à l'interprétation, ici, les cas positifs pour les 2 gènes sont considérés ainsi pour la valeur de cycle seuil inférieure à 35. En termes de résultats, ici, par rapport à la distribution en termes de nombre d'échantillons, au total, nous avons eu 5 échantillons, dont 15% de positivité. Et en termes de distribution par an, il faut remarquer que chaque année, on trouve quand même un cas positif, dont 3 en 2021, 2 en 2022 et 4 cette année. En fonction du type du gène détecté, nous avons trouvé pour le gène Pool A 9%, alors que le TP représente 8% des cas positifs détectés. Alors, selon le gène détecté et par année, nous remarquons que seul en 2022, que le gène TP est en proportion inférieure par rapport au gène pool A. Sinon, en 2021 comme en 2023, ce sont les mêmes proportions de cas positifs qui ont été détectés. Alors, ici, il s'agit d'une distribution en termes de lieu de résidence des patients. On constate ici que les cas de PIAN ont été détectés en 2021 et en 2023 dans le district de Meaux, dans la région centrale. Un cas a été observé cette année dans le district de Thun et seulement en 2022 dans le district d'Antwerp. On note que depuis la confirmation par les tests de DPP en 2019-20, aucun cas n'a été confirmé dans le district et dans la région des plateaux. Alors, en conclusion, nous pouvons dire que la PCR nous a permis de confirmer la présence de Treponema pallidum dans les districts, notamment endémiques, confirmés par les DPP, dans le sud de Meaux et de Thun. Et ces données peuvent être un soutien au programme de maladies tropicales négligées dans son processus d'éradication de cette maladie. Alors, quelques perspectives en vue. Nous pensons faire la recherche d'autres pathogènes pour faire la thèse de différenciation, notamment l'hémophilus du CREI. Et puis, par rapport au programme, il s'agit d'étendre les investigations à d'autres districts et enfin, réaliser le séquençage génomique afin de voir la transmission et les caractéristiques génétiques du pathogène ainsi que sa transmission dans le pays. Voilà ce que j'avais à partager. Merci beaucoup à tout le monde.
Merci. Merci à Dr Isaka pour la présentation. Comme toujours, nous sommes prêts pour les questions. Michael, you have the floor.
Firstly, I can congratulate you. It's great to see another country bringing PCR online for yours. I think broadly speaking, the results you show are what we would expect from the literature. You know, if you look in Ghana or Côte d'Ivoire or Cameroon or in the Pacific, only about 25% of serologically confirmed cases will the lesion contain treponemal DNA. And that, of course, is because you can be asymptomatically infected with yours and have a different cause of your current skin lesion, right? So you can truly have yours, but it just not be the cause of the ulcer on your skin at the moment. So you will get a separation between your serological result and your molecular result. So I think the results you show, I totally believe. Secondly, I'm really pleased to see that you have adopted using the POL-A target. I think this is a good example of why we need to harmonize the TP0858 gene, which I know you used originally on its own. This was published by the CDC in America. I will not be too rude about the PCR on a live webinar, but in my opinion that PCR is not usable. We have demonstrated clearly that it has primer binding site mutations, and you see that in Togo that you have a POL-A positive, TP0858 negative. I'm sure if you sequence that, there will be a primer binding site mutation in that gene. For example, in the Solomon Islands, every single yours sample we have ever tested is negative on the TP0858 gene, developed by CDC, but by whole genome sequencing is confirmed to be yours. So I think that's your data from Togo shows exactly why the LabNet needs to harmonize the procedures to make sure everyone is using the best performing PCR. So really just congratulations. I think it's wonderful data, and I'm really so impressed to see Togo getting that off the ground. I think it's a huge step forward for the yours program, both in Togo and across sub-Saharan Africa.
Thank you, Michael. This is a suggestion. Congratulations. Other question?
Okay, we have no question. Thank you, Dr. Isaka. We are calling now the next speaker. The next speaker is Dr. Valérie. In my program, Dr. Valérie. She's coming, yes. You have 10 minutes to present degrees of newborn ulcer case experience from 2 major endemic health districts in Cameroon. 10 minutes, please.
Bonsoir à tout le monde. Bon après-midi. Je vais faire comme mes prédécesseurs et je vais aussi parler en français. Donc, je suis Valérie Duncan. Je suis la responsable de l'unité de mycobactériologie du Centre Pasteur du Cameroun, qui est le laboratoire national de référence pour la tuberculose et l'ulcère de Boroli. Donc, je vais vous présenter ici l'expérience du Cameroun pour confirmer la baisse de la détection des nouveaux cas d'ulcère de Boroli. Donc, ok. Vous avez ici sur ces 2 premières figures. Sur la première figure là, on nous montre la baisse de l'ulcère de Boroli, de nouveaux cas de l'ulcère de Boroli dans le monde et au Cameroun. Ça, c'était entre 2014 et 2002 et 2014. Et ici, nous avons cette baisse-là qui est notée à 54,8% entre 2010 et 2017, dans le monde également. La troisième figure ici, c'est le Cameroun avec les différents sites endémiques d'ulcère de Boroli. Et en 2021 et 2022, nous avons mené une étude sur la recherche intensive des cas d'ulcère de Boroli dans les deux zones endémiques, notamment à Konolinga et Ayos. L'idée pour nous d'avoir mené cette recherche active des cas en 2021 et en 2022 venait du fait que pendant la pandémie Covid, dans nos différents pays, nous avons bien noté qu'il y avait une baisse à la prise en charge des malades ou la surveillance de certaines pathologies, dont l'ulcère de Boroli. Nous nous sommes dit donc en 2021 et en 2022, après la Covid, d'aller faire cette recherche active des cas-là pour voir si effectivement, normalement, en fait, on devrait avoir la réapparition ou bien l'augmentation des cas de ces pathologies-là après Covid. Donc, nous sommes allés sur le terrain pour pouvoir confirmer est-ce que les cas ont réellement augmenté parce que la prise en charge avait baissé ou bien est-ce que ce n'était pas le cas. Donc, nous avons ici les deux principales zones endémiques, là, Konolinga et Ayos, où l'étude a été menée. L'étude s'est faite entre juillet et août 2021 et en septembre 2022, où nous avons inclus dans l'étude tous les cas présomptifs d'ulcère de Boroli que nous avons trouvés dans ces zones endémiques-là. Donc, pour réaliser cette étude-là, nous avons travaillé avec les chefs d'aire de santé, les agents de santé communautaires, les préleveurs, les techniciens de laboratoire également. Donc, comment est-ce que nous on était organisés? Au niveau de l'aire de santé, le responsable d'aire de santé était chargé de sensibiliser les agents de santé communautaires sur la reconnaissance, l'identification des personnes qui sont suspects ou présomptifs des cas d'ulcère de Boroli. Et une fois que ces agents de santé communautaires étaient sensibilisés à l'identification des cas présomptifs d'ulcère de Boroli, ils descendaient donc dans les communautés. Et en communauté, ils avaient la charge d'identifier donc ces personnes qui sont présomptives des cas d'ulcère de Boroli et de remplir le questionnaire ou le formulaire d'enregistrement qui avait été fait à cet effet. Donc, une fois qu'ils avaient identifié les cas présomptifs, ils travaillaient en étroite collaboration avec les préleveurs. Les préleveurs, une fois informés des cas identifiés en communauté, faisaient des descentes en communauté, autant pour moi, où ils devaient, à la suite de l'agent de santé communautaire, confirmer ces cas identifiés-là, faire le prélèvement. Avant de faire le prélèvement, ils obtenaient d'abord le consentement éclairé de la personne. Le prélèvement était fait et le questionnaire rempli. Donc, une fois que tout était OK, le préleveur devrait amener les échantillons au niveau du laboratoire dans le district de santé. Et c'est à partir du laboratoire donc que les échantillons devraient être transférés au centre Pasteur du Cameroun où la PCR était réalisée pour le diagnostic. Nous avons ici dans ce tableau les principaux résultats et nous pouvons noter ici que nous avons eu en 2021 et 2022 15,6% des cas d'ulcère de Buruli, donc 12,4% en 2021 et 20% en 2022. Et les personnes les plus atteintes, c'était des hommes âgés entre 36 et 55 ans. Et je vais également dire ici que les personnes, les cas présomptifs d'ulcère de Buruli qui ont été prélevés avaient déjà commencé un traitement traditionnel. Donc, nos résultats obtenus en 2020, à 2021 autant pour moi et 2022 sont inférieurs à ce qui avait été obtenu avant au Cameroun dans les mêmes sites. Nous avons eu en 2009 29,4%. Là, c'était dans un autre site. Mais en 2016, on a conduit une étude à Konolinga. On avait eu 27%. En 2019, on avait eu 42,4%. Dans la baisse des nouveaux cas d'ulcère de Buruli, nous pensons pourrait s'expliquer, comme l'étude a été faite après Covid, par le fait que les mesures de barrière qui ont été mises en place pendant Covid pourraient avoir participé également à la baisse parce que les gens ne se rencontraient plus beaucoup, les gens se lavaient les mains. Donc certaines mesures d'hygiène qui ont été mises sur pied pourraient être à l'origine de cette baisse. Ou bien les efforts combinés mis en place avant par les ONG, par les programmes nationaux et par les projets de recherche, peut-être ont commencé à avoir leur effet. Ou encore, nous on a pensé que les interventions menées sur le terrain il y a plus de 10 ans, comme la réhabilitation des puits, l'utilisation des moustiquaires, le nettoyage des alentours des maisons, le port des habits à manches longues quand il faut aller au champ, et le port des chaussures aussi pourrait être pourrait expliquer la baisse des nouveaux cas d'ulcère de Buruli. Donc pour cette étude, nous avons eu quelques challenges sur le terrain, notamment 3. Le premier challenge, c'était l'accès à la communauté parce que ça s'est fait pendant la saison de pluie et avec le mauvais état de route, ce n'était pas évident d'atteindre la communauté. Un autre challenge, c'était la conservation des échantillons une fois au laboratoire parce que l'idée derrière, ce n'était pas seulement de faire une QPCR, mais c'était également de mettre des échantillons en culture. Dans les échantillons, dans les laboratoires des hôpitaux de district, les mesures de conservation des échantillons n'étaient pas remplies. Et aussi le transport d'échantillons. Donc dans ces zones reculées-là, on n'avait pas d'agence de voyage spécifique pour pouvoir effectuer le transport des échantillons. Donc il fallait à chaque fois assigner quelqu'un du laboratoire pour pouvoir transférer l'échantillon au Centre Pasteur du Cameroun. Donc, comme conclusion, nous pouvons dire que nous avons eu 15,6% de cas d'ulcère de Buruli à Ayos et à Konolinga en 2021 et en 2022, et que ce pourcentage est inférieur à ce qu'on a eu les années d'avant. Donc, ceci pourrait confirmer qu'il y a vraiment baisse dans l'apparition de nouveaux cas d'ulcère de Buruli dans ces deux régions endémiques du Cameroun.
Minute.
J'ai fini. Merci.
Merci, Dr Valérie, qui a présenté des résultats qui montrent la baisse de la transmission de l'ulcère Buruli. S'il y a des questions, it is time now for questions. Docteur, vous avez la parole.
D'abord, je félicite plutôt l'équipe du Cameroun pour le travail. Ma question, je ne sais pas si entre-temps j'ai été un peu distraite lors de la présentation, mais par rapport à la conclusion, comme quoi il y a baisse du cas d'ulcère de Buruli basé sur la décroissance ou la baisse de la proportion de positivité. Est-ce que vous vous êtes posé la question de savoir si entre-temps la définition de cas de Buruli même n'a pas varié entre ces années-là? Ça, c'est la première question. Et cette variation ou cette modification de la définition de cas peut faire en sorte que la définition devienne plus sensible en termes épidémiologiques, c'est-à-dire une définition qui ratisse tout ce qui est comme ulcération, et de ce fait, vous aurez une proportion de positivité faible. Voilà essentiellement mon inquiétude, c'est une seule inquiétude en fait. Donc si vous pouvez nous clarifier si entre-temps il n'y a pas eu une modification de la définition de cas. Et aussi l'équipe qui collecte les échantillons. Est-ce qu'entre-temps il n'a pas changé? Il faudrait qu'on soit sûr que la définition de cas a été maintenue dans le temps pour que cette proportion de positivité puisse vraiment traduire une baisse du nombre de cas. Merci.
Docteur Valérie, vous pouvez répondre?
Oui, par rapport à la première question concernant la définition des cas d'ulcère de Buruli, la variation avec le temps. Bon, je connais pas la définition des cas en 2019, en 2009 autant pour moi. Mais je sais que pour l'étude, nous avons utilisé la définition des cas la plus récente définie par l'OMS pour pouvoir identifier les cas d'ulcère de Buruli. Bon, maintenant, est-ce que ça a varié depuis quelles ont été les variations depuis 2019? Ça, je ne sais pas. Peut-être ceux qui travaillent dessus depuis 2019 pourront m'aider.
Je vais peut-être donner la parole à ma voisine, parce que je sais que pour Buruli, pour confirmer un cas, il faut avoir une PCR positive dans tous les pays. C'est ce qu'on a donné comme standard. Pour confirmer Buruli dans un pays, il faut faire la PCR. Je ne sais pas si tous les pays appliquent, mais c'est ce que l'OMS nous a dit en 2019.
Identification.
C'est la définition clinique. Si nous sommes partis sur une définition qui est trop sensible, c'est-à-dire qui ratisse trop large, nous aurons une proportion de PCR positive très faible. C'est ce que je veux savoir. Est-ce qu'entre-temps, ça n'a pas changé sur le plan clinique? Ça n'a pas varié. dans votre pays ou bien lors de l'étude?
Merci. Ce que je pourrais ajouter, c'est que nous n'avons pas utilisé le score, donc on n'a pas modifié la sélection des cas. Et je voudrais aussi dire que, en fait, quand on était allé sur le terrain, on nous avait dit qu'il y avait beaucoup de cas du fait que pendant Covid, on ne pouvait pas pris en charge et que même avant Covid, le programme MSF s'était arrêté, donc il y aurait beaucoup de cas. Donc on a travaillé avec le programme, on a réuni les healthcare workers, les agents communautaires qui travaillaient déjà sur le site de Bourouli pour les resensibiliser et voir les zones où il y avait... donc en fait, on partait en se disant qu'il y aurait beaucoup de cas. Ils nous ont dit que tout c'était chez les praticiens, donc on a fait une sensibilisation en amont. Donc on espérait, non, mais on pensait qu'on aurait vraiment beaucoup de cas, mais le score qui permet déjà de bien définir avant n'a pas été appliqué dans ce cas-là. Donc c'était vraiment, c'est vrai, un peu moins sensible parce qu'on prenait tout ce qui était les plaies, c'est quoi la durée, c'est au moins 5 semaines ou je sais pas, donc voilà. Donc en fait on partait parce qu'après Covid on se dit, et déjà avant Covid il y avait déjà eu un arrêt un peu de prise en charge, et après Covid on avait dit il y avait plein de cas, donc on en a profité pour dire on fait une recherche, active case research.
En off, ils vont encore discuter en off. D'autres questions? Allez-y, docteur.
Oui, je me joins à la première préoccupation de la collègue. Donc, dans la présentation, l'équipe docteur Valérie aurait mieux aussi présenter les faiblesses de l'étude. Et ça, par exemple, ce qu'on vient de citer là-bas, ou en termes de couverture, et balancer maintenant avec l'intitulé de l'étude. Parce que j'ai vu en anglais, vous avez parlé de confirm. Est-ce que vous êtes à l'aise avec les potentielles faiblesses de l'étude à parler de la confirmation de la baisse?
Les faiblesses de l'étude n'ont pas été présentées pour faire les conclusions qui vont avec les résultats qu'elle a trouvés. Donc, est-ce qu'il y a eu des faiblesses dans cette étude-là? C'était quoi les faiblesses? C'est ça, il veut remettre. Si le docteur Valérie peut répondre.
Oui, c'est ce qu'on vient de dire là. Il est juste en train de dire que ça devrait normalement figurer dans la présentation les faiblesses de l'étude. C'est bien compris, merci.
Docteur Issa, oui.
Yes, thanks. Actually, my question is a little bit the first question from Jacqueline, whether the case definition was the same, but also if the experience of the clinical teams were similar, or maybe there was a decline in the suspicion rate. My understanding is that since MSF and FAIRMED left these areas, the clinical expertise has been declining, so maybe that could be an explanation. No? The clinical suspicion is not as specific as before. And also, if because of COVID-19, maybe many people attended the traditional medicine, and maybe some of the approaches of traditional medicine, they will not heal Buruli ulcer, but maybe they will have some impact on the bacterial load, and that's why try to consider these things.
Maybe.
Allez-y.
Oui. On n'a pas la confirmation de ce qu'il est en train de dire, mais comme l'étude a montré que la plupart de nos cas présomptifs étaient déjà en traitement traditionnel, il s'est dit peut-être que le traitement traditionnel a eu un impact sur la charge bactérienne et du coup était efficace. Ce sont juste des suppositions pour la baisse. Mais comme notre prédécesseur a dit, quand on a travaillé uniquement dans les deux zones endémiques, peut-être pour confirmer qu'il y a la baisse, il faut aller dans les autres zones pour faire un peu dans toutes les zones et puis comparer. Mais ce qui est sûr, c'est que les études menées déjà avant au niveau du programme, le programme national contre l'ulcère de Buruli montre déjà cette baisse-là au fil des années des nouveaux cas de l'ulcère de Buruli au niveau du Cameroun. Peut-être c'est la raison pour laquelle nous on a parlé des cas confirmés, la baisse confirmée.
Si, je comprends mieux aussi. D'autres questions? S'il n'y en a pas, je pense qu'on va applaudir Dr Valérie pour ce travail difficile. Nous appelons maintenant Dr Sundip pour présenter. We are calling now Dr. Sundip for the presentation of advancing point of care molecular diagnostic for skin negligent tropical disease. You have 10 minutes, doctor.
Good afternoon. I'll be quickly talking about advancing point-of-care molecular diagnostics for skin-neglected tropical diseases. This is not new for some of you who are already involved in these studies, but I just wanted to give a quick update on what we are doing with the BU-LABNET qPCR and the projects that we are involved in. I'm Dr. Sandeep Chaitanya Vedidi. I work as the Senior Director of Research and Innovation at American Leprosy Missions. I also work as the Senior Research Associate in the Department of Medicine at the University of Cambridge in England, currently positioned at Cambridge. These are some of the pictures of our experiences on the laboratory bench, bedside, and the community with this particular technology that I'm going to talk about. BU-LABNET qPCR, I got in touch with this company. Before getting into the details, I declared that we don't have any direct conflict of interests, personally or professionally. We got in touch with this company in 2019 when they were presenting at Colorado State University on their work with the armed forces in the United States, actually helping the armed forces to detect diseases in their camps. Yes. Oh.
You need translation, please.
Right. Can I?
It's working now, the translation, please. For French people.
Okay.
It's okay now?
Should I continue? Or?
The batteries are down. All this. So I can't hear it.
Wants to be given to the people who need translation, please. I can help collect.
Can I continue now?
You can start now?
Yes.
Yes, okay.
Okay. Can you get the French translation? At all? No. Yes. Okay. Right. So we got in touch with this. Yeah. We got in touch with this company in 2019. Since then, I have encouraged them to invest their technology into developing assays and solutions for neglected tropical diseases, especially skin neglected tropical diseases. While we interacted with them, they also demonstrated some experience of working in Africa with some of the viral diseases. They managed to develop assays for the viruses, and it's been in the space since 2015, but it came into light more in 2017 and 18. Later, the company has grown vastly during the COVID time. I'll just quickly talk through the technology. DNA extraction is basically through a simple cartridge system. This cartridge will be on my desk if you would like to see. There are 6 chambers in this. So since many of us are molecular biologists here, each chamber has got, the first chamber has got the lysis buffer, second one more of DNA wash solutions, and then there is an air-dry empty chamber where you just have an air-dry process, and then the elution. So there is a syringe there that you can see on the slide. Syringe has a column which is fitted here, and DNA can actually be trapped in the column here, and then the wash steps and the elution continues. So you add sample in the first chamber. It should be a liquid sample, so you need to homogenize if it is a biopsy. We have tried with blood, we have tried with skin, slit skin smears, extracted, directly introduced into the first chamber in 1x ppb or double distilled water. Swabs can be directly inserted into the first chamber, which has been tried in BU. And then you put the syringe in, you just pump in and out, and the last chamber, you get the DNA eluted. We tested it with 2 mycobacterial species, both leprosy, Mycobacterium leprae and Borrelia Mycobacterium ulcerans, with our colleagues. Some of you are already here. Michael is here, who has done this extensively. The process is about 2 to 3 minutes, and the quality of DNA is good for PCR amplifications. Just yesterday, we heard from Wendy as well that she managed to follow the same protocol and managed to break the grains in mycetoma. Solution is the cartridge is packed, so cross contaminations are very less likely. From our experiences, we haven't seen any cross contamination so far. PCR reagents are in a strip-based system, so you submit your primers, probes. You can actually optimize the whole PCR in your lab for any species, any gene. You send the primers, probes, and the concentrations to the company. Company will lyophilize this strip and will send it back to you. So we managed to manufacture both BU as well as leprosy assays. They call them assays, 3-well ghost strip assays. The capability of the machine is 3-plex, so it can read up to 3 genes in each single well. PCR is a small brick, as you can see here. This is the full-blown real-time PCR. This is just a miniaturized version of the full-blown PCR. There is no artifacts in terms of the technology. There is a quencher on one side, there's an emitter on one side, and quencher on the other side, and it has got 9 wells. So the throughput is low, throughput is 9 wells, but you get the whole run done in 50 minutes. I'm just giving these machines to some here, so I just brought them with me. Software is a mobile phone app, which is, I just opened it for people to see. You can just run on any phone, either Android or Apple. And then when you come to an area where internet is available, you can actually, the data actually gets uploaded to the cloud portal. Cloud portal is a secured portal that's sold along with the machine to the user, and user can actually log in and see. And user can add up to 10 additional users now. So dissemination of results in a cloud portal is quite feasible with this system. We tested it extensively on the bench. That's the picture I would like to always show. We tested it at the bedside in India for leprosy, especially with the center in Vellore. Few weeks ago, I took the machine into field, Songololo in DR Congo. Delphin is here who supported the whole process. We managed to confirm 2 cases of BU, suspected BU, with ulcers, directly taking the swab samples and doing the PCR right beside the patient at a primary health facility. Initially, we started this work with the goal of actually evaluating the whole system. If we could reproduce what we see on the laboratory bench in this miniaturized model. So we tested for RLEP and IS2404. Our collaborators in KCCR, NMIMR, Naguchi, and CPC have done the Buruli ulcer work, and I did the leprosy work back in India with our colleagues. This is our presence in the beginning. When we started off in 2019, we have put the machines in these places and developed for both for leprosy as well as for BU. Now we are spread across. There are a few more countries that I need to add to the list. One will be Côte d'Ivoire, that will be receiving a machine in the next half an hour, which is right beside me. And also Erasmus Center and others that will be brought onto the list. As I said, we started the development process. So it involves 3 different things. Development, originally you need to optimize the PCR in the lab, submit the PCR format to the company to manufacture assays, bring the assays back and develop, do an evaluation in comparison to standard PCR to really say that these assays are working. We did all these 3 with BU. We did the first 2 in the sense development and manufacturing for leprosy. We still need to do a rigorous evaluation. And then we slowly stepped into the multiplex mode, trying to use the system to multiplex for 5 diseases. The purpose is not to really look for co-infections, but co-endemicity, to address co-endemicity. If in a co-endemic situation, if one panel is available for all these 5 diseases, one could even break those tubes and straight away use them in the machine rather than waiting for reagents to be procured for each disease. So that's the goal. The third project, the multiplex project, was just recently initiated, and ISWAD stepped into support, and Foundation Rolf Laro added, and American Leprosy Missions also support this initiative. We partner with labs in endemic countries. Would you like to move this down? Yeah, that's great. We partner with labs in NTD endemic countries. We test biomeme qPCR on clinical samples, standardize and develop qPCR assays, and validate those assays against the TPP use cases. Buruli ulcer, this is the sensitivity and specificity we have seen. Actually, it's achievement of our colleagues, Michael Frimpong, who has been instrumental in developing the whole piece. Assays are manufactured. They are available with us. Some of them are with Estil and I think also in KCCR. So you can, if you would like to test, if you would like to run some tests, please contact any of us. For leprosy, we just got the strips a few weeks ago. Some of them were distributed to the labs. Some were still with us. If somebody needs to evaluate them, please give us shout out. We have seen a sensitivity of around 96% and specificity of 100%. I'll be breaking down by paucibacillary and multibacillary later in the slides, but this is the current situation. For leprosy, while doing the developmental piece, we looked at the genome equivalents, the cells. At Karigiri, they have the dedicated animal house facility for Mycobacterium leprae propagation in the footpads. So we took those cells, we enumerated them, and then we did a PCR just to see if genome equivalents match with the CT values, as you can see in the left there. And then my colleague Charlotte Avanze at the Colorado State University has actually developed this plasmid with RLEP gene in it, and then did serial dilutions of the plasmid. Then we took some samples at Karigiri, which are actually collected from another study, and then tested them for biomeme, on the biomeme platform, just to assess the CQ values. We did leprosy cases, household contacts, and endemic controls. From all of them, we took slit skin scrapings. For contacts and controls, it's just earlobes because we don't have any patch or an indication. But they are endemic controls from a very high endemic area. You can see that the CQs could be differentiated between the three groups, but more data is needed to really conclude anything here. With Buruli ulcer, we did this, KCCR did this cross-validation, KCCR, CPC, and NMIMR. Initially, at KCCR, the correlation seems to be 100% between the standard real-time PCR and the biomeme system. I think these slides were shown, so I'll just quickly skip them. M1 preps also showed good sensitivity and specificity. In 2 centers, there were low sensitivity for some, around 75% for some reason, which we evaluated later, but at least KCCR managed to see it more than 92% sensitivity. Comparison of standard qPCR with biomeme. As you can see, the CQs at all the 3 centers or CTs at all the 3 centers seem to be slightly lower than the same sample on a regular routine machine, which indicates that the signals produced by this machine are not compromised. These are some of the key observations. We have seen that it works equivalent or superior to the standard qPCR. The most advantageous thing is the lyophilized assays. They are quite easy to ship, very simple to carry around, and we tested it in the community recently, making it reproducible in the context. I talked about these new projects. They're just kicking off. The second one is still in discussions. We are talking with ILAB partners. Some of the ILAB partners came forward to commit funds, but we need more to really do this rigorous evaluation of the leprosy assays. The multiplex project, as I was telling, is now starting off. These are the countries involved for now, but I think we may expand once we have the original concept proved for multiplex assays. For the evaluation, these are some of the centers that we have listed based on the geographic distribution and also representation of countries with high endemicity for leprosy. Here we have 2 phases. The phase 1 is the multicentric evaluation, which we will be doing in the following countries. Phase 2, we would really like to assess the feasibility of implementing at the periphery of the health system. This is just a representative diagram, but this is something we would like to do, to test the same sample at all the 3 points with 3 different setups and personnel to see if there can be a reproducibility. And if the CQs are nearly reproducible, that's when we would say that we have taken the technology to the field. Thank you very much. And this is the picture of the child who has gone through BU and got...
Thank you, Dr. Sandeep. We have now time for some questions. I propose 3, 4 or more, okay? Question? I see Michael.
Thanks, Sandeep. I mean, it seems like lovely technology, and it was Sandeep and I went to visit the biomeme offices earlier in the year with Michael, and the performance seems excellent. I mean, because basically it's qPCR in a very small machine, so we should anticipate it to be good. I think thinking forward to the sort of multiplexing aspect, we'll need a bit of thinking. Because, for example, there are 3 channels on the machine, so there is obviously a natural limit to the number of things that you can multiplex. For example, mycetoma alone will be more than the machine can have, but it's more than most qPCR machines can do, 3 or 4 channels. So I think we will need to think about multi-stage processing. For example, you have an ulcer, you put in a strip that has treponema pallidum, cutaneous leishmaniasis, Buruli ulcer, it tests positive for treponema pallidum. You put in a second strip that has the 3 targets for drug resistance. Similarly, one can imagine the same kind of stages. So we need to be innovative in the way we think about that, and also recognize that the shopping list of multiplexing will vary for different countries. What Professor Fahal will want in Sudan will be slightly different than what someone else will want. So I think it's very promising, and we just need to continue to think about how to optimize the combinations versus the single plexes, recognizing that both may have use.
Thank you for the statement. I think just to quickly respond to it, for mycetoma, as of now, what we thought was, if we could use this to differentiate just actino and eumycetoma, that should be the good starting point. I think Wendy can add more, but that's where we are. And just to clarify on the multiplexing, it's 3 genes per well. So if you're just taking one sample, you can do up to 27 pathogens in the whole panel. 3 9s, 27. Yeah.
Dr. Sarah?
Know about the price,
Okay.
extraction price, the machine,
Sorry, I somehow missed it.
the strip.
Yeah. This is currently $5 per test. We are talking with the company to bring it down to $3 per test if they can. The machine is approximately 10,000, but we are trying to talk with the company to see if they can still continue to offer us a good more discount, because if it is a single plex, it's 5,000. If it is 3 plex, it's you can adjust the plex. They don't have to come and do any repairs. They have a lock code with which they can remotely open and close your plexes. So, so if you'd like to. Strip currently is $7. It might reduce further, but at this stage it's $7 a strip.
Doctor, thank you for the answers. Continue the question. Isra?
Thanks, Sandeep. Questions regarding the format. So you say the machine has room for 9 tubes.
Yes.
Do they come in a single strip of 9 tubes, or the strip is 3, 3, 3, or something like that?
You asked the right question. So this is a completely open system. You can run just one tube. They have 3 well strips, basically 3, each one is 3 wells. You can use 3 well strips. You can use a 1 9-well strip completely if you can break it, because they also have plates.
Okay.
So, and we have tested normal plastics on this machine with other reagents, Eppendorf and other reagents. It all worked really well. So it's
Okay,
an
so-
open platform. They can.
So
you can
then
do it.
we could combine in the same run, we could combine tubes with primers probes for different diseases, and actually the multiplexing capacity will be higher than 3 detection. No? Another thing, as a qPCR machine, is it possible to do melting analysis if we don't work with probes and we work with intercalating agent like cybergreen, something like that? Is it possible to do melting curve analysis?
Melting curve analysis, yes. It has high resolution melt included just recently.
Okay.
So this version of the machine now has it. Actually.
So then that will even increase our possibilities.
Yes.
Last question. How does it comes now? It comes dried reagents without primers, I understand. For example, if we want to start working on lathe, should we order to the company that they add the primers, or we add the primers wet, or how it works?
Company has 2 formats. One format is total molecular biology reagents. So they will sell the master mix and bulk reagents, which you can directly use. You can use your own primers, probes, set up a PCR on the bench, similar to how you do. And then when you send these primers to them to lyophilize and create assays, they will create, and then they will send those assays in batches to us. 2,400 batch samples a batch.
Yeah, the price will be similar to what you have explained before, no?
Yes, the bulk reagents are less pricey, but if you manufacture the assays per tube around $7.
Okay, thanks.
Thank you. Other question, comment, suggestion? I don't see. Okay. We thanks Dr. Sundip. And we invite now the last speaker for this research session, Professor Lydia Mossi. I give you the floor.
Yes, I can sit here because it's a discussion, right? Or I have to go to the front? Do I have to go to the front?
It's not discussion. You can sit. You can sit.
I can sit here and be more comfortable. Suffering is optional, isn't it? Okay. Well, thank you so much to the organizers of the meeting for including a session on research collaboration. It is a discussion, so more of the feedback or comments will be coming from the general floor, and I am just supposed to be like a shepherd, you know, heading the discussion, not the people. So I've been told by my boss Numfor that the main idea of this session is to target the discussions to see possible collaborative studies that all the labs represented here can be part of. And I'm actually very glad that Professor Phillips presented earlier on today about the research topics which are of key importance, not only for Borreliosa but for other skin NTDs. And he beautifully teased out 4 major thematic areas, which I think we can sort of adopt as our collective action plan going forward in terms of collaborative research. So a quick summary of, or should I say a quick reminder of these 4 areas were disease transmission, which includes active case surveillance, epidemiological modeling, and adopting technology in mapping and other innovations that can collectively advance the knowledge in disease transmission. The second one was on diagnostics, and this includes case confirmation, community-based case surveillance, and something that I want to slide in here, just because of what we've been seeing and hearing in today's meeting, about is it time for us to look for another molecular target for diagnosis, or is IS-244 still the best guy? I don't know. And 3, case management. The topic or the idea of piloting new treatment strategies as and when they come up. And again, something I want to slide in here, the untapped potential of herbal medicines or local remedies that people have used over the years and it has worked anecdotally. And the possibility of increasing educational campaigns in so many diverse forms, either by cartoons or by movies or by documentaries or whatnot, docudramas, to help with disease prevention strategies based on what we currently know about what the possible risk factors are, especially here in Africa. And then finally, the need to still do more anthropological work to uncover the true socioeconomic burden of the disease and the ripple effects that it has on households and the mental stability of affected persons, the stigma and discrimination associated. So these are the 4 thematic areas that Prof. Phillips beautifully elaborated earlier on this morning, to which I've just added a few snippets. So I guess to start off with the discussion, I want to think about it in 2 main ways. Currently, what are the resistors that we have in place that are preventing us from working together to address these concerns? First thing that comes to mind is technically lack of funding, right? That's one of the resistors. But is it possible to identify other possible resistors? So I think we'll start with talking about what the resistors are generally. What are our challenges? Why is it that we've not been able to work, even within country, when there are 3-4 research groups all working on Borreliosa or working on skin NTDs? Why haven't we been able to come together to work together, or to apply for main research grants together, and so on and so forth? When we exhaust the list of resistors, I would like to invite us all to think about what are the assistors. And to start thinking through what our assistors are, I think when we look around in this room, we are our own assistors. We have a fantastic network of people who are on the field, who understand how difficult it is to get a sample or how to facilitate getting a sample. We have people who are in the labs who are well trained on how to analyze the samples when they come. We have professors here and doctors here who are capable of putting our findings into research publications. and can even help with the docudramas and so on and so forth to communicate the science that we are finding. We are our own assisters. And even more importantly, we have the LabNet, which provides the network or the platform to which we can even assist each other more and stop working in silos, but rather at a large scale, even to apply for a grant, okay? And also, another assister is the fact that we have national control programs in almost all the countries that are represented here. How can we improve the activities of the national control programs? How can they help, for instance, in getting ethical clearance for our research activities? How can they facilitate transportation of samples from the health centers, which are in the very rural areas, using the national grid or the national programs that they have to transport samples to research centers or research labs? So when we look at the assisters and we look at the resistors collectively, we can come out with how might we's, which are how might we solve these problems and how might we address these problems in terms of research, in terms of championing our research activities. And then finally, maybe we can talk about who can fund all of these activities, okay? There are so many calls there. I know World Bank is really interested in driving African centers of research excellence. Is it possible to have a network of laboratories or research centers which are collectively an African Center of Research Excellence for skin NTDs, just like you have an African Research Center for infectious diseases, and so on and so forth. So in my mind, this is what I've thought about, and I hope this brief introduction has sort of teased our thinking caps and woken us up from lunch a bit to contribute to come up with some ideas by which we can drive our research collectively. So I guess the floor is open now, and if there are any suggestions, I'm happy to take them and steer the discussion. So, yeah. I guess it's just a raise of hand and then we call. Or should we start with the resistors? Because it's easy for us as human beings to think about the negatives. So when we talk about disease transmission, what are some of the resistors, and so on and so forth. So yeah, I've named a few, so we can build on these.
Maybe I'll just...
Thank you, Mark.
I think, as we've said before, the main barrier is that no matter how much we would like to set the agenda, much of the agenda is set by the funders. And so I think revisiting that idea of like taking the points that you have made, making them concrete and working out, having a shopping list so that when specific calls come up, the network can activate to respond to those calls. The network is a very strong resource, but we won't be able to just go out and say, well, we want money for transmission. But what we can do is develop a list and, you know, 1-page, 2-page concept notes that collectively the network can address, such that when appropriate calls become available. That's thing one. And thing two, I like your sort of shout out for the World Bank. We also have to be realistic that many of the NTD funders do not care about this particular group of NTDs. And we must just accept that, I think. We are never going to win the Gates Foundation over. I have given up on that battle. And we must think about other funders. There are other funders out there, the World Bank, MasterCard Foundation. You know, obviously we've been very lucky with the leprosy foundations, but there are alternative funders outside of the main funders who are looking for spaces to occupy. And so I think we need to be more proactive about thinking who are the alternative funders beyond always the EU, the MRC, that we can approach.
Thank you for that. I saw Solange turn on her mic first. I'll come to you soon. Ladies first. Ladies first.
Nous sommes déjà à cette réunion où on doit parler de la recherche. Et Michael Max vient de dire quelque chose de très important. Il faut de l'argent. Donc nous devons mettre des notes conceptuelles. Qu'est-ce qu'on veut faire? À qui on adresse cela? Parce qu'il y a des fonds, mais il faut qu'on fasse comme on dit, il faut qu'on fasse des choses pour attirer tous ces bailleurs. Donc moi, je suis d'accord pour la transmission, c'est la recherche. S'il n'y a pas d'argent, il n'y a pas de recherche. Donc c'est ça que je voulais partager en français pour vraiment réitérer que c'est nous qui cherchons l'argent. Donc il faut produire des documents qui attirent de l'argent à des bailleurs qui en ont. C'est par rapport à la transmission. Parce que ce n'est pas leur priorité, la transmission, c'est le diagnostic, souvent. Je vous remercie.
Thank you. Thank you so much.
Thank you very much for the opportunity to speak about this important issue. There is a good body of literature talking about international research collaboration. In fact, now there are some systematic reviews about the difficulties, about the paper published on the international collaboration and difficulties and constraints facing the researchers. I think one of them is the culture barrier. There is a lot of culture changes and behavior changes between different communities, local and international, between the north and the south to the south. So I think we need to know the culture in the country where we're going to do our research. We need to know the value in that culture and that community. We need to know how we can target the community where we're dealing with. I think this is one of the important barriers for doing a proper international collaboration. Some of the international collaboration, they want to get only biological material from you, and that is it. And to be published in a good journal, and that is it. But they don't look into the community affected and what will be the importance of that research on the community at this side. The important thing that the culture of working together, the teamwork. Many of us, they don't work in teams. They don't know how to do these things. And we have to respect each other. We have to know what is we need from each other. We have to make sure the patient is the center of our research. If the patient is not in the center of that research, that is not a good research. That is for publications, put in a good journal, and that is it. And I think we always think of the patient at the center of all these things. We have to look into culture of things. We have to look in the values of the communities we are dealing with. Even for the researchers themselves. I believe in something, you are not believe in that. So you have to come in compromise about doing things, something at this side. And I think if you can manage, we have the language barrier, we have geographical barriers, and you have to overcome all these so as you can have a proper consortium formulation, and then from there we can go forward for doing our research. Thank you very much. So I mean the social dimension of this collaboration should be respected and should be observed. Thank you very much.
Thank you so much for that contribution as well. It definitely is very paramount, and it's actually the fourth thing that Prof. Phillips mentioned earlier on today, the need to really conduct socio-anthropological studies, not just focused on the country that you're working with, but what if it's a different whole dimension that is occurring in Cote d'Ivoire versus Ghana, and so people are predisposed to risk factors differently, and so on and so forth. It's really true. Are there any resistors, any more resistors that we can think about so that we can collectively come up with assisters? I know this time is really hard to think, but we need to think. Especially for those on the field, those who have to, those who are involved in active case surveillance. For me, that's one of the biggest barriers, right? Active case surveillance, even how to get samples regularly. One of my challenges currently in my research group is there are some funded projects that when they go looking for samples, they incentivize the healthcare workers so much that now if you yourself go with peanuts, because you can't come up with the same sort of incentive, the attitude to helping you do active case surveillance becomes a problem. Okay, so how might we make sure that we are not turning active case surveillance into a, I don't know, a marketing opportunity or a promotion or whatnot. It's a big challenge for me personally, because you can only write so much into a grant to use as incentives, right? It's one of the challenges that I'm having. I don't know how other countries are able, especially Benin. I think Benin is one of the countries that has an excellent surveillance system. So how can we learn from what Benin has done? Yves, you want to say anything about Benin? I know you've not been on, but yeah,
I'm
you're
from
even here.
Benin.
Exactly.
What I can say is medical personnel is very included in the collecting of the samples. Medical person, national programs, working together is very important. Laboratory members have to regularly share the results with the national programs. That includes the program into the work that we made. And the information have to pass through all members, not only with the lab members, but around lab members, medicines, community workers, and national programs. That is, I don't want to, how to explain it. It is the work together. It's collective work. Then that is what I can say. Yeah. Perhaps Dr. Barogi can add something else. I don't know.
I want him to talk, so please talk.
Thank you. I have experience from Benin, but I don't want to speak as a representative of Benin here, but I just want to share the experience I know about Benin, because you know, Benin has a good community-based surveillance. I think it is a key aspect. And in Benin, we don't give money to the community health volunteer. We have another motivation for them, like give them a motorbike or something like that. So it is a specific organization in Benin. And as he said, we usually work as a team. So at the community level, we have a community health volunteer who suspect the cases. After that, we have a health worker at the peripheral level who supervise this community health volunteer, and they can try to make the first level confirmation. And at the intermediate level, we have a reference health center who can collect the sample and send to the lab. But I think before all these teams work together and we know exactly what they are doing. So it is summary what I can share with you. Thank you.
We appreciate sharing that with us.
Please.
Yes, please.
I can complete by saying that, for example, in the center of Bobe, we have some kind of team of volunteers workers, and when they check one case, we have a network, and they take the photo, sent to the doctor to say, there is one case here. And per week, we have 2 days, and medicines, doctors goes to the village to check the cases. Then the doctor comes to the village to see what volunteer workers said, to take the sample to confirm it, and brought the sample to a laboratory to confirm by qPCR.
Yeah, I think that's a fantastic system, which for me, I think is now an assister. And we identified a resistor, and we've just found out what a good assister is. I will include that in the notes at the end of the discussion as a possibility to sort of increase our case surveillance actively. The other thing which again we've seen in some of the presentations, is how technology has been adopted in mapping out areas for where you have hotspots of the bacteria and how you can superimpose patient cases on those hotspots. It's a very nice multi-layer way of identifying how transmission is progressing within a village or within a community. So I think maybe for those who are interested in transmission research, this is something that can be seminal in whatever proposal that you're coming up with, so that we can have a more concrete case evidence of where the cases are and where the genotypes of the microbes for these diseases are. I'm going to move quickly to diagnostics again, because it's the next thing that the next thematic area, and I can already say that the BU-LABNet has been fantastic at improving diagnosis, even with the EQA that we are doing. I mean, it's something that is not easy to do, because when you get your results and you realize that maybe you have 2 false positives or 2 false negatives, it's like a wake-up call that you need to be doing something better. But I think it served as a fantastic assistant to normalize or harmonize the kind of work that we are doing. What I'd like to get feedback from is our thoughts about harmonization of protocols. This morning I came in a bit late, but in the discussions that were going on, one of the things that I noticed is that maybe something can be developed to perfection in Belgium, but just humidity or temperature would change its response in Ghana. So how might we improve on these harmonization protocols? Should it be only one group making the harmonization protocols, or should there be more or protocols that work for different geographic areas, which would be shared and discussed further, and so on and so forth? It's just a thought to drive the discussion. I see Dr. Yango looking at me intently. I don't know whether it is sparking something in her mind. It's just a thought. I'm leading a discussion.
No, before, perhaps I didn't understand well, because harmonization of procedure is going hand in hand between partners and local lab. So if we're talking about Buruli ulcer, we did it only on local lab. Nothing was done in Belgium. That's why I was looking for
I just used it as an example. I'm not saying that that's what has happened.
But even currently, if I take the example of yours, there was no research or evaluation in London, but Michael, as expert, was working with us, and we did it in Yaoundé. And now we were looking to see which lab are also volunteers, so that they can also validate to see if they get the same results before harmonization.
Yeah.
That is how we work for Buruli ulcer too. So, for example, if we take mycetoma, so we need Wendy first, because she's the expert on mycetoma, and sometimes we did not have all the experience with us, so we need them start. So for leishmaniasis, I call a negotiate team, but I think they were not ready to get in, so we start with Javier to do not lose our time, but we know that we cannot harmonize or only what Javier found in Spain. Even if the WHO Collaborative Center, we have also to test it in the country. So there is no harmonized procedure, just validate outside of our country.
I'm not saying anything different from what you're saying.
Okay.
All I'm saying is, so for instance, I use Eppendorf equipment in my lab. And I'm saying, for instance, in my lab, let's say I use Eppendorf equipment for everything. like an epinephrine thermocycler, epinephrine whatnot. And in another lab, they use biorad. So when I'm talking about harmonization, I'm going into the nitty-gritty of even equipment and so on and so forth. And whether we need to consider that, it is part of the discussion, not to say that we are doing something different. But whilst we are thinking about harmonization, do we need to think about all of these? That's just the question I'm asking.
Okay. So I think at my level it is difficult to think also about harmonization of equipment. What we are trying to do is to see that the protocol we are putting on can work in different machines. That's why we are trying, even for yours, when we validate for BioRide, since we have 3 or 2 different equipment for PCR in our center, we are trying to test different equipment to be sure that it could work, because it will be difficult for us to tell to the other lab that only run it by RAD, only run it by, if you cannot give them the equipment. If we are ready to give the equipment, so you can say that, okay, use the one we give to you. But what we are trying to do is have really a procedure that we can, in open machine, we can apply it, whatever the equipment you have. So I know we can think about it since we have issue or challenge with equipment maintenance, if you can talk about this maintenance around the country, but you know that is also difficult. And like we discussed this, I think, the first day, and it's like a commitment of our government or our institution for maintenance. So I think it's really difficult at this end to say that we can also harmonize equipment. Yeah. That is the other people can also give the point of view.
Yeah, thank you. Very well appreciated. Very, very well appreciated. Did you want to say something, Sandeep?
I think this has been an ongoing debate in several networks and several laboratory networks as to how to really harmonize the whole system. I think in the context what we are talking, that may not be really feasible right away, but the procedure is the procedure. So if we are able to do a protocol properly, if we are able to capture the CTs with efficiency, I think the harmonization of the protocol and the reagent should do a lot than the machines themselves.
Yeah, I agree. Mark? Well, Michael?
Maybe I'll just try and steer us back a little bit on topic, away from harmonization of SOPs, which I think was this morning, and back onto how we can do better at collaborative research. And I guess I'm keen for Lydia to be given some concrete actions that we will take. I think, of course, harmonizing SOPs is good, understanding how we can improve sample collection in the field is good, but what are the concrete steps that the network will put in place so that over the next 1, 2, 3 years, the network itself will develop and submit grant applications to embed research within the network? And I think that is the ask for us, right? So that it is not me at the London school coming and saying, can I use the network for my project? But it is the African leadership of the network identifying, not in this room, we're obviously not going to fix that, but what are the 1, 2, 3 priority questions that the network would like to address? Who would, even as I say, just having concept notes of what those types of ideas might look like. I think if we're to go from a sort of broad statement that we would love to do more collaborative research together to a concrete action plan, probably within the network, someone needs to take a lead. It doesn't have to be at the coordinating center, but within the network, someone needs to say, we're going to survey across the network what are the research priority questions that the network specifically is uniquely placed to answer, develop that shopping list of ideas, and then task people with developing outline proposals. So that's what I would really encourage you to do. I think for the leaders in this room who lead the network to go away and think, if I'm going to put 3 grant applications in over the next 5 years, what are the questions that I think that the network can answer? Who would be the partners in those? What would be the questions? I think that's what you need to make something concrete, because otherwise a grant call will come next year and everyone in the network will say it would be great to apply, but...
Yeah. Well, thank you for bringing us back to topic, Michael. I guess a follow-up to that will be, where can we undertake these kinds of research activities that we are thinking about? Should the research be done by all the labs? Are all the labs involved, or only in the research? I don't know. So it's a question again that I'm putting out, because I remember from last year, even just speaking with other people in the network, there are more than one health center or more than one group here that wants to be part of the research. But as to how they can be involved in the research is the question. So how do we bring everybody on board, and who does the research? Where is the research done, and so on and so forth? It's something that we need to talk about. Oh, yeah.
Sorry if this point has been made, because I've been going in and out and I just came in, and so pardon me if this point has been made. I think, I don't know, but as it is now, it will be difficult to say there's a call, all of us are responding or not being part. I think one of the things that we could do is to develop sort of few questionnaire to know which institution is a part of this network, what are they doing, what are their capabilities, what is there. Because if the institution you add and they don't have the capabilities, then they cannot be part of it. So I don't know whether this has been made. If that has been made, then pardon me. Other than that, I think it's a very important exercise within this discussion that, for example, if there is transmission, is the institution already has expertise in doing this transmission? If it's genomics, does the institution has the capability? So we need to look at that, if we can do kind of survey. Thank you.
Fantastic input. Thank you so much for that. That's what we were asking, yeah.
And I think those things will tie together, right? If you generate a list of priority questions from that, for question 1, maybe it will be yourself and Sarah and Isra will be the people that are interested within the network. For question 2, it will be Dorothy and Eves and Wendy. You know, I think by having your short list of questions, the different network members will be able to say, yes, I'm interested in this question, I have the capability. I think these things will flow together. Sorry, I interrupted Richard, which is bad of me.
I was going to make a point on similar lines, because we need to know the capabilities within the network. Of course, we do know each other, but probably unless you've been to my center and maybe you get to know it better, you will not know exactly what the capabilities. But we need to know what capabilities we have within the network, because if you need to form a consortium, you need somebody with this scale, that scale, that scale. Who do you see? Sometimes calls come out, you need someone from Benin, you need someone from Cameroon. Quickly, maybe Sarah will come to my mind because I've been working with her for a long time. But we need to know each other better, know each other's capabilities. Once we know that, when there's a call and you are putting a team together to sort of apply, you know who and who can lead, and potentially the network can get people to work together. So knowing those skills is very important. Expertise within the network is key to us moving forward. Apart from having the shopping list that Michael has said over and over, we need to, because previously when we met, we did have an idea of some papers that we wanted to do as a network, as a start. I mean, a sort of review article. I think we did put something together previously. Do those points stand? Were we able to achieve them? Are they low-hanging fruits, that areas that can get us to start doing things together? Are they good reviews and good? We could easily come up with desk reviews that this consortium we can initiate and put together to stamp our authority on the scientific landscape. I mean, it sort of helps us to work together, and as we work together, we get to know each other better. Bringing people from different parts of Africa to work to, instead of always the same people doing the same thing, we'll get them to know each other better, and then the network becomes more vibrant. So I think the communication lines is also another thing to help us to know each other better. Okay, thank you.
Prof, you have the floor then.
Thank you. I speak in French, please. Non, depuis hier, Kingsley et nous autres, nous avions déjà proposé qu'il y ait un tableau qui va présenter la situation globale au sein du réseau, où on peut même l'appeler checklist, qui présente les différentes institutions qui sont là, leur capacité technique, je sais que ça a été fait au départ, et je pense qu'il est question maintenant de faire une mise à jour, et on aura même les ressources humaines pour savoir où c'est disponible. Et puis, ce tableau-là pourra être partagé tout au sein du réseau pour qu'on puisse s'y référer, et même en dehors du réseau pour se faire connaître. Je pense que l'équipe qui a travaillé sur la communication pouvait avoir pensé à cela. Donc, c'est ce que je voulais ajouter par rapport à ce que Dr. Philippe vient de dire. Merci.
Thank you so much. And I also think it also includes our collaborators. If we have other collaborators that are not within our current network, who we think can help us, there's nothing wrong with also bringing them aboard on the research, not necessarily the BU-LABNET.
Okay. Dr. Lydia, le temps, you are looking for the time. Donc, je vous donne encore peut-être 5 minutes parce qu'il faut qu'on tienne aussi compte du temps. Merci.
Okay. Thank you, Prof. I want to speak to what Professor Richard just spoke about concerning working together within the network. I mentioned it twice. The first time in Cameroon, the second time, even at the last meeting in Cameroon, that Nigeria is always left out. Whenever there was any call in the past, I would reach out to Numfro and Dr. Sarah to help look for interested partners within the network. And the call, maybe 1 or 2 months to respond before deadline, nobody will show interest in collaborating with Nigeria. If we look at the presentation so far, Nigeria is left out. And we have been, Nigeria is a member of, even founding member of this network. I always reach out to a few people that I know that could link. There was a time Professor Fahel from Sudan also reached out to the network. I doubt if anybody apart from me responded to him. It's not really encouraging. When you see presentations, you always see the same set of people reaching out to the same group, you know. It's not really the best, and it's not encouraging.
Okay.
Yeah, we can.
That's hard.
We can
That's
add, we can add our voices to that. Yeah. So, if we really want to collaborate, we should be able to go beyond people we know. Working with people we don't know is also equally important. You feel comfortable because I know you before, so there's a tendency that if anything, I will call and talk to you so that we work together. But that doesn't strengthen the network we would like to establish. So I think the points that have been made should be noted for going forward. I remember this NSBAD support, I had to say that no, you have to put Sudan there because mycetoma must be there. So though Sudan is not in the network as we speak, but if you want to work on mycetoma, it's a must, you must involve Sudan. Whether we've worked with them before or not, it doesn't matter. And we should look forward to doing so. By involving other labs, we also build capacity because we can't always be the Noguchi, the KCCR, the Pasteur Center, etc., leaving DRC, leaving Gabon, leaving Liberia. How will they also move on in the network? So I will challenge all of us to do better on this one. We can't sit here and talk collaborative research when it is only among the few people.
Thank you, Dr. Siedu. I think technically our time is up, but I just want to summarize real quick what has come out from this. One that stands out very much is that we need to get a shopping list almost of what is or what are our key issues that we want to address in terms of research as a LabNet. Not necessarily BU-LABNET, but as an SNTD LabNet. We need to come up with a shopping list. We've already gotten a fantastic insight of some of these in 4 thematic areas, which can be broken down further. So maybe after this, we can suggest sitting down and preparing a Google Doc that can go round, where we put all of these things in there and see which labs are interested in what. The second thing that comes up strongly is that we need to, again, do another shopping list, but this time is a shopping list of our areas of expertise. Not only areas of expertise in the lab, but our human resource or people on the ground who we can mobilize to assist with all of these research collaborative ideas. The third thing that comes up strongly in all of this is to identify a list of funders who we think we can sort of approach when we have our concept notes and so on and so forth. I know for a fact that some of these funders are even ready to work with researchers, especially a collection like this that has a proven track record. I mean, there's even a publication from BU-LABNET, which is fantastic preliminary data of collaboration within the network, which we can use as a stepping stone, and they can help us from concept notes up until the end of a full-blown proposal. And paying attention to this African Centers of Excellence scheme is something that we can tap into very, very easily. And then finally, the thing that I probably want to ask of Kingsley, since he's mostly approached by some of these funders, is to help tailor their want to give money to the areas on our priority shop list and not just anything out there. So these are some of the summaries that have come up from the 30-minute discussion we have. I really want to thank you all for chipping in and making this very interesting. Thank you so much, and for the opportunity.
Thank you, Professor Lydie. We give now the microphone to Dr. Sarah. If you have some comment for this discussion. Thank you.
Wow. So thank you very much, Lydia. Kingsley, we well noted your comment. Professor, we noted your comment. But sometimes we are really busy, and when someone just sends you that there is a call, yeah, you noted that there is a call, but there is not a concept note or something to invite you to work together. I think we are working with Professor Fahal. We have submitted one call, but it didn't work, but we continue to work, and we really consider that we cannot work or think on mycetoma without him, without Wendy, and now we bring up Senegal team. So we really noted our shopping list of expert teams, shopping list of topics. Please, when we also send Google form we have to insist during 2-3 weeks, sending 10 emails before we get also the answer. So that is our baby now, and we have to all take care of our baby, so we have to take time, give our time so that we can continue to grow together. So really, thank you all, and we have noted all the comments, all the suggestions, and we will try to do better, and we will engage to be better, but you also, you have to continue to work from the development of our network. It's our baby. Thank you.
Invite everybody to the break, coffee break.
There's a coffee break.
Yes, it's coffee break now. 20 minutes max, 20 minutes. And we come here for the general comment, for the closing. Okay, thank you.
Oh, dear.